Hi everyone, we're into week 5 and now we're starting chapter 7. This chapter deals with membrane structure and function. One of the most critical aspects of cellular function involves this exchange and communication across the plasma membrane or across other membranes inside the cell. A lot of our pharmacology, druggable targets are on the membrane surface. Things like AIDS, HIV, COVID, all deal with membrane structure and function. So let's dig into it a little bit, shall we? Without a plasma membrane, cells are just chemicals floating in a larger solution. And so having this ability to have what we call regulated exchange between the external environment and the internal environment is absolutely critical. Now, you might have heard this term before, and it's selective permeability. I like to say selective permissibility. That's because most substances just can't make it through the membrane on their own. Now, certainly some gases can, non-polar molecules, things that are hydrophobic. Some water can squeak through there, but most of the water that gets through is actually permitted through using protein channels. And so it's good that we can keep things in that we want to keep in and keep things out that we want to keep out. But there is a caveat. There is a downside to that we have to deal with. And that is lots of things that we might want in can't get in on their own. And we're going to talk about that. Well, I mean that they can't get in on their own because they're just too water soluble. The selective permeability, the permissibility, has to be modified to allow these molecules in. And we're gonna show you how we overcome those barriers. Another key concept about a plasma membrane, and especially a plasma membrane, it's not untrue with other membranes, but the plasma membrane is very true, is that it is a mosaic. This is a photograph of a mosaic from Roman times where you have different pieces of material that they use as, it's like a pointillist picture where you have different pieces of material creating a larger framework of an image. And that is equally applicable to the fluid membrane of a cell. It has different kinds of phospholipids, it has different kinds of proteins, different kinds of molecules that give that membrane unique characteristics and functional ability. And just for a moment, let's contemplate an image like this, which shows a membrane in cross-sectional profile, along with an outer surface rendition and an inner surface rendition showing cytoskeletal elements. This is a microtubule and intermediate filaments and microfilaments. It's showing you the mosaic nature. And unfortunately, as beautiful as an image like this might be, it's hard to learn. You kind of have to build up from the basic components, and that's what we intend to do in this chapter. We're not going to be that busy when it comes to these models because it's just too much to take in. We are going to get after the different components just in lower abundance, shall we say. So the first thing we're going to deal with are the phospholipids. They are sort of the mortar in the plasma membrane. They are amphipathic. That is to say, they have a hydrophilic side and a hydrophobic side. We've talked about that before. You will find other lipids in the plasma membrane. You'll find fatty acids that anchor proteins to the membrane. You'll find cholesterol. In any case, though, phospholipids are the most abundant of the lipids. And the fluid mosaic model says that you have these lipids, and they have lots of lateral motion, but you'd be amazed. So we know from doing kinetic studies that these lipids can move sideways very quickly, but they can't flip. They can't flip from the inside to the outside. So it's very stable in this vertical plane, but it's dynamic in the horizontal plane. And so if these lipids can't flip, then neither can these proteins. But those proteins are embedded. And that's what we mean by fluid mosaic, is that we have this lateral motion that makes it like a fluid, but it has other things embedded in it, just like a mosaic might have different colored tiles in different places. Now, in terms of your guys' perspective, 1972 feels like a long time ago, but in the grand scheme of science, this is actually a pretty new idea. It's only 50 years old. Put it in perspective, Niels Bohr and Albert Einstein were doing their thing in 1900, 70 years earlier than that. So this is even though it is getting to be a little bit far away, it's getting farther away every year. That's how it goes. It's still a relatively new idea, and it's been revolutionary. The last, in my lifetime, there has been an amazing explosion of knowledge about the plasma membrane, and this model was proposed by Nicholson and Singer, and it holds true today. The evidence seems to indicate that this is the valid model. I'm leaving this picture here only for your reference. I think this is a good picture to come back to from time to time to help you get your bearings on the outside of the cell, the inside of the cell, and the proteins and other molecules where they're found. One thing that we're going to have happen as we move forward in this class is that we're going to show you examples of evidence. Rather than just tell you what is, if you know what I mean, say this is the plasma membrane, we're going to actually try to show you pieces of evidence, and that's an important transition to make in your undergraduate careers from a strictly this is this, this is this, this is this, to why do we think this is? What is the rationale behind ideas? If you can pick up on that skill of defending your rationale with your evidence, you're going to be way ahead of the game when it comes to undergraduate education. So here's an example of some evidence. What they did was they took a cell and they embedded in this special sort of polymer. It's a type of polymer that they use for electron microscopy. And they very, very carefully fractured that polymer so that what happened was when they peeled off one piece of it, they only took 1/2 of the plasma membrane. They were able to separate what we call the leaflets. They have or layers here. They say extracellular layer and the cytoplasmic layer. So they were able to extract that and take a look at the imagery. What they found was on the inside of the extracellular layer, they found these bumps, and those are proteins. You can actually test for that. But on the cytoplasmic side, look at the difference. Look at how many proteins are there. And that's saying that there are a lot of peripheral proteins and partially embedded proteins on the inside of the plasma membrane, and a few on the outside. There's still quite a few there, but nothing like on the inside. So we know that the layers actually differ significantly. But the idea of this fluidity is a lateral fluidity. These things can move sideways, but they can't move vertically. They can't flip from leaflet to leaflet. So the lipids may be some proteins if the proteins are not anchored to any other structures. If they're anchored to a cytoskeleton or they're localized in what we call a lipid raft, where we have a sort of an enrichment of a certain kind of phospholipid, the proteins will stay relatively still. If they're not tethered down, then they'll drift around laterally. But rarely, and this is rare, so we know that from doing high-speed camera and what we call photo bleaching. So what they'll do is they'll use a fluorescent marker on the phospholipids, and then they'll hit it with a laser beam, and it'll bleach out an area. And what they'll do is they'll watch the unbleached molecules move into that bleached area. You can actually see lateral diffusion going on. It's really, really cool. But in any case, we see this happening 10 million times a second. So if you see an exchange between one or between two phospholipids, swapping places happens 10 million times per second, but one flipping from the inside to the outside is at once per month. That is trillions of fold in terms of orders of magnitude. So it's very, very stable on the vertical plane and dynamic in the lateral plane. Pretty. Here's another experiment like the photo bleaching one. What you have is you're going to create what we call a hybridoma. You should write that down, hybridoma, because what you do is you take a mouse cell that has membrane proteins with a certain color marker, and you take a human cell that has a different color marker, and using the right techniques, you can get them to merge. We call this a hybridoma. They don't last very long. They eventually separate again. But what we have observed is that when we create this fusion product, we can observe the movement of the lateral movement of proteins. After an hour, you can see a significant mixing of the red and blue colors. But that's called a hybridoma. And this is just a picture that I found doing research looking at different membranes in a cell and the different head groups found on the phospholipids. Like PC means phosphatidylcholine, or PE, phosphatidylethanolamine, phosphatidylinositol, phosphatidylserine, and so forth. And you can see that these compositions differ based on the type of membrane and the function of that membrane. So these head groups are all different kinds of head groups on the membranes in the phospholipids. Another aspect of the fluid mosaic molecule is how fluid, how apart are the head groups. And there's what we call a homeostatic window for the right amount of distance between the head groups for just the right amount of permeability. If we have a membrane that has lots and lots of unsaturated hydrocarbons, so they have like cis double bonds, especially cis double bonds, like maybe one here or maybe even two in some cases. In any case, when you have that membrane is very, very separated in the head groups, which means it's very, very fluid. On the other hand, if you have lots of saturated fatty acids, that means they have all single bonds in here. The membrane has the opportunity to become very, very densely packed, very viscous. This may be too, on the left may be too fluid, and this one on the right may be too solid. And that's where we have to find that in between. Oftentimes we find these membranes rich in unsaturated fatty acids in colder environments, in organisms that live in colder environments, like for instance salmon. Where you get those omega-3s, those fatty acids, those omega-3s, that's because they live in colder environments. We also see them in plants too. But we have to have that right amount of viscosity. Too fluid, we don't have regulated interaction between the inside and the outside. Too viscous, and now we can't get anything inside and outside. So about as fluid as salad oil is the general rule of thumb. And a lot of work has been done in this area, in part to understand why tissue and organ systems fail when it gets too cold or too hot. We have heat stress and we have cold stress. And so understanding those may give us a better handle on how to deal with things like, for instance, frostbite or hypothermia, or hyperthermia and heat stroke, those types of things. Because if we can understand fluidity here, we can understand how to keep cells alive. And so they look at what we call the shift between a solid phase, a gel-like phase, and a liquid-like phase. But one of the key molecules, is cholesterol. It's a steroid. We've talked about that before. At warm temperatures, cholesterol restrains movement of phospholipids. So above 37 degrees or at or around 37, it'll keep phospholipids together. But on the other hand, at cool temperatures, it'll maintain fluidity by preventing tight packing. It serves as a fluidity buffer. And cholesterol intercalates right in between the fatty acids. There's a little OH there. That little OH just sticks out right in the end here. It's a little bit aqueous right there. Helps keep it in place. But that cholesterol helps keep the phospholipid heads just far enough apart on average to maintain proper fluidity even in the face of warmer temperatures or cooler temperatures. And lots of data has shown us that variations in cell membrane go hand in hand with the kind of climactic zone that a plant or an animal will inhibit. This map right here is actually a guide for showing you what plants will do well in different zones and what plants will not do well in different zones. And that has a big part of it has to do with the constituency of the plasma membranes of those plants and and by extension animals. And that's a big issue when it comes to climate change. Too much change too rapidly, and animals and plants can't adapt quickly enough to meet the environmental needs that they face in their given area where they live. Now we're going to switch to the membrane proteins. Certainly phospholipids have a role to play when it comes to function and permeability, but it's the proteins that really drive function. Proteins are the doers in cells, and so we have membranes that are partially or completely embedded in the membrane that have lots of different functions, and we're going to break those out. But first of all, we need to talk about location. Location, location, location. Now, we will classify these things, but the truth is it's a spectrum, and I'll explain what I mean. So first of all, we have what are called peripheral proteins. These guys are just around the head groups or maybe just a little bit inside the head groups, just barely penetrating the hydrophobic area. We call those peripheral. If they penetrate deeper, like maybe a little more than this one, more than halfway across, we could call them integral proteins or integral proteins because they are really penetrating into the hydrophobic area. And to be able to do that, you have to have hydrophobic regions within the protein. You have to have amino acid chains that have lots and lots of hydrophobic side chains on it. If you span all the way through, we call those transmembranes. So transmembrane proteins are part of the integral protein family. How these proteins are created and how much hydrophobicity they have determines how embedded or all the way across they are. A protein just doesn't make its way across a plasma membrane. It has to be inserted, usually in the rough endoplasmic reticulum is where that happens, right, during synthesis. But if there's a hydrophobic region, it may just self-associate. But you have to have those hydrophobic nonpolar amino acids in order to achieve that. When it comes to function, your job here is to be able to recognize and explain what each of these functions are. What I will tell you is that proteins will often have more than one function. For instance, a protein might be involved in signal transduction that may be involved in anchoring or movement of ions or materials across the plasma membrane or enzymatic activity. That oftentimes we have more than one function in a given protein. But the 6 categories of function include transport, moving things across the membrane that wouldn't otherwise move on their own. Enzymatic activity, we localize metabolic enzymes. A classic example of this is the mitochondrion. But if you look at a bacteria, the inner surface of the bacteria is loaded with metabolic enzymes because they don't have any organelles. Signal transduction, the communication of information from one side to another, often involves these others. Like for instance, if we want to know there's another cell nearby, that might involve cell-cell recognition, and then that causes a response. Or maybe we're talking about neurons where we're sending information using ion flow, and so forth and so forth. So transport, right here. Enzymatic activity, right here, as there are enzymes that are embedded in the membrane. Signal transduction, information flow, changing the mode of the signal. Like for instance, these yellow guys never make it into the cell. These yellow chemicals never make it in, but nevertheless we send information down the line. Cell-cell recognition. This is very, very important in the immune system, but also important for proper patterning of tissues. Joining of cells, holding them together, making sure that they don't slide against each other and they have tensile strength or compressional strength. And then attaching to the cytoskeleton and the extracellular matrix. But again, there are more than one function for a lot of proteins in the membrane. And these are the figures from your book, so you should be able to recognize these. And we're going to try to come up with examples for each of these for you. That's the goal here, is to see what these things really do in cells. And here are the other ones. Intercellular joining doesn't look all that fancy, but these other ones are looking pretty good. Again, that cell-cell recognition, the attachment to the extracellular matrix, and other tissues. We're going to start with cell-cell recognition. How a cell recognizes who and what another cell is and what that cell is doing. Did you know that you get cancer all the time? But the incidence of diagnosis is less than 1 per lifetime, and that's because of cell-cell recognition. Turns out that when cancer cells develop, most of the time, the vast majority of the time, they start expressing proteins on the surface that flag them as not normal. And your immune system comes along, sees that, and says, destroy that cell. In fact, a lot of cancer risk occurs because of immunosuppression. That cancer somehow finds a way to evade the immune system and evade this cell-cell recognition. The membrane protein that is most involved in cell-cell recognition are glycoproteins. These are the name tags, if you will, of a cell. A protein embedded in the plasma membrane that has one or more carbohydrate groups attached to it. We also have glycolipids out there, so we have phospholipids that have carbohydrates attached to it. Remember that carbohydrates are on the exterior surface of the cell, so this is in between two cells. Cytoplasm here. We're just in extracellular fluid out here. So those carbohydrates, the only carbohydrates you see in any significant concentration inside a cell are simple sugars like glucose, or ribose sugars, and glycogen, which is a polymer of glucose. Other than that, there are very, very few carbohydrates inside the cell. Most of them are either polymerized in glycogen or expressed on the outer surface, particularly in animal cells. Starches and other things, that's a different story. But for animal cells, most of those carbohydrates are external. And I have this moving GIF here for you, hoping this works. But what it's doing is it's showing you this contact, this interaction. These glycoproteins are interacting with each other and causing a response inside the cell. You remember how I said that cell-cell recognition can interface with things like signal transduction? Well, this is an example of that. If you have a cell recognizing another, it means that there has to be something going on inside the cell. So you can see that a lot of studies have been going on here about understanding about how these cells recognize each other. By the way, this interaction right here is critical for determining appropriate tissue donors. If you have an inappropriate donor, what will happen is the white blood cells in the recipient will attack that tissue and cause it to be destroyed. This complex right here, this immune synapse, is actually a glycoprotein complex interacting on either surface of the cell that helps to recognize self versus non-self. A really fascinating example of how important cell-cell recognition can be has to do with HIV. It turns out that HIV, like a coronavirus as a matter of fact, has to bind certain proteins on the surface of a host cell. For instance, it's a receptor protein called a CD4, and this other protein, the glycoprotein called CCR5. These are receptors, and they're localized to the outer surface of the membrane. With a coronavirus, it happens to be a membrane enzyme called ACE, or the isoform 2 of ACE, ACE2. In any case, with HIV, it has to have these proteins in order to bind and penetrate the plasma membrane of a host cell. That's why HIV can only infect certain cell types. It can only infect CD4 positive white blood cells and certain neurons. Those are the predominant forms of cells HIV can invade, which is why you get immune suppression. The viruses come in, they destroy these CD4 cells, which coordinate cellular responses, and the person comes down with an immune deficiency syndrome. Latent immune deficiency syndrome, or if you see a baby that's been infected at birth with HIV, you'll see neurological issues where the virus is invading neurons in the central nervous system. But there are people in this world that don't have CCR5. They lack CCR5, and so they are immune, functionally immune to HIV. The HIV can't penetrate the membrane, and thus they cannot get HIV. We've actually cured HIV because we accidentally gave bone marrow from a CCR5 negative donor to a cancer patient, a person with leukemia who had HIV. They killed off all of the bone marrow in the recipient patient and then gave the donor marrow, and that person's HIV went away because it could no longer infect those cells. Problem is, though, that CCR5 does a lot of other things too, and so people who don't have CCR5 are vulnerable to other types of sickness. And this was also the site of controversy. There is a new type of technology called CRISPR-Cas9, and a Chinese scientist claims to have taken identical twins, and one of the twins, he said he knocked out CCR5 in that twin. And so these twins are born now, and I haven't seen much follow-up on that, but that scientist has disappeared because hugely, hugely unethical. At least right now, we haven't determined the appropriate ethics for using CRISPR-Cas9 in human cells or intact human beings. But in any case, his idea was he could make one of the twins immune to HIV. We're going to get to the other functions in a little bit, and especially into part 2. But the reason we did that cell-cell recognition first was to emphasize that idea of the sidedness of the membranes. That when we manufacture these membranes, they are manufactured in an asymmetrical fashion. That they have distinct and inside and outside faces. The outside is rich with carbohydrates, some proteins. The inside is much more rich with proteins, but you don't see any carbohydrates. So again, be able to determine which side is which based on where the carbohydrates are located. But we make plasma membrane in a structure called the Golgi apparatus, named after the person who first identified it. We've talked about Golgi. You have this cis Golgi where vesicles come in, and trans Golgi where vesicles come out. But the Golgi is one of the sites where we manufacture new plasma membrane. And the Golgi are polarized just like the plasma membrane is. So for instance, if you have a vesicle come in, inside the Golgi is a hydrophilic interior. Okay? The outside is cytosol. Notice that once we merge with the plasma membrane, that hydrophilic interior becomes part of the external environment. It's a lot like your digestive system in that respect, that as you pass from your mouth down through to the other end, you have external environment going through you. And that's what's going on here. And the Golgi is one of the major sites for synthesis. And what we see is that vesicles, and not all the vesicles have cargo in them, the vesicles will migrate and fuse with the plasma membrane. There are other times, though, that when plasma membrane is too much, they'll actually take up vesicles in order to reduce the amount of plasma membrane. We've already talked about this concept already, but that's going to set us up, this idea of permissibility, selective permeability. We'll only let things through that we really want, unless they have special characteristics. Some things can get through that are beyond our control, but a lot of things are under control of the plasma membrane. That's why I say permissibility. Before we get to those transport proteins, we need to talk about what molecules can get through a membrane and what can't. If they're lipid soluble, if they're small and uncharged, they can usually get through the membrane quite easily. Examples include gases. Oxygen, carbon dioxide can move through the membrane quite easily. Very small molecules that are lipophilic, like nicotine, for instance, or another drug that we use for motion sickness called scopolamine. Again, small molecules, very, very hydrophobic. You can put a patch of nicotine right on your skin and it'll pass right through the plasma membranes of all those cell layers and get into the bloodstream and be pharmacologically active. But if they are not lipid soluble, if they're water soluble, if they're charged, or if they're larger, they need help getting through the plasma membrane. In this case, we have an ion here, and we have a channel that allows movement of this ion. We're going to talk about those transport proteins in a little bit. So as I mentioned previously, if the molecules are really, really small and uncharged, they can move through the membrane through a process we call simple diffusion. The cell really isn't regulating this other than perhaps regulating the phospholipid content of the membrane. But by and large, this is a relatively unregulated exchange. Gases, very small lipophilic molecules. If a molecule needs assistance getting through the membrane, like for instance, let's say glucose over here, these little hexagons, or charged molecules, like if we're doing something for signaling or something, we have channels and carrier proteins. This is another channel over here. It's a special one. So transport proteins in the form of channels and carriers allow hydrophilic substances or charged molecules, sometimes slightly larger molecules, to cross the membrane. If they're truly large molecules, we have to use vesicular transport. So channel proteins are like protein tubes that allow, usually ions are the most common thing. One exception would be aquaporin. Aquaporin is a type of protein channel that facilitates water equilibration, water diffusion, osmosis. Water diffusion is osmosis, so we can move ions with channels, very, very small molecules. Generally, channels are selective by size and by electrical charge. Aquaporins are selective for water, and carriers, they're more specific and much more like enzymes, but they actually change shape. Channels don't change shape except to open and close. So here are channels on the left, and here are carrier proteins. And what you see with a carrier protein is it's a lot like an enzyme. You have a binding event, a change in conformation, and then a release on the other side of the membrane. Channel proteins, some facilitate diffusion like protein channels do. Others actually move material against their concentration. We call that active transport. So here's from your book. Here is a channel protein. Again, mostly very, very small charged molecules. Channels are selective for size and charge, and they facilitate diffusion. So there's no energy input here. On the other hand, carriers, like I said, are like enzymes. They bind, they change conformational shape, and then they release. Some of these ones will facilitate diffusion and are passive. Other ones will move against a concentration gradient and require energy, usually in the form of ATP. We're going to stop right there for now. We're going to finish up Chapter 7 in the next video. So thanks for your time. As always, if you have questions, please let me know, and I will be more than happy to help you in any way that I possibly can. Thanks again.